So, just to brush up, elicitors induced effects in plant cells include effect on calcium metabolism, which means because calcium is used as a second messenger and can play a role in transcription of certain proteins which may involve their participation in the secondary metabolism. So, therefore, calcium metabolism can indirectly affect the secondary metabolite biosynthesis. Now, there are reports where there are set receptors in the plasma membrane of plant cells where these elicitors bind to. Once they bind, there is a signal cascade mechanism which can lead to cyclic AMP formation, and this cyclic AMP burst leads to further calcium ion concentration increase in the cytoplasm, which then binds to proteins, thereby then getting into the or affecting the transcription of certain proteins responsible for secondary metabolism. So, massive variations in membrane integrities might happen because of elicitors. Protein and phosphate metabolism might happen. Then ethylene, so when I say protein or phosphate, protein you can directly make out, and even with ions, because the channels or the permeability of the membrane may get disrupted, because of which the electrochemical gradient can get disrupted, and because of the change in the ionic concentration, maybe the pH, can lead to further enhancement in the secondary metabolism or direct release of reactive oxygen species. And differential gene expression, where gene expression responds to signals or triggers. So, now consequently forming enzymes concerned in the synthesis of polysaccharides, which mean plant cell wall enzymes, or hydroxyproline rich glucoproteins in the cell walls via induction of proline hydroxylase. So, these are some examples by which different types of elicitors have been found to play a role and induce secondary metabolism. Then polyphenolics which get deposited in cell walls may get altered. Then chitinases, these are certain enzymes or PR proteins, if you remember when we had studied, it might get altered, which play a role in secondary metabolism. Or specific proteins against pathogenic infection, this is what is PR proteins, phytoalexins. Now, mechanism of action, further removal of regulatory repressors, genetic manipulation of the enzyme itself, which means that their expression level can get changed, which may be involved in biosynthetic pathways as metabolic inducers for increased secondary metabolism. Then they can provide stimulus, which is the basis for exploiting the biotechnological potential of these plant cells. Then when we say stimulus, stimulus means in the form of overexpression of the genes or the encrypted genes, because of the repressors they may get induced. So, these are the different ways. So, what are the different? These are just example enzymes which get altered by the calcium ion concentration in the cell. NAD kinase, then NAD oxidase reductase, protein kinase. The protein which I was talking about where the calcium gets bind, these are generally kinases. Then calcium transport ATPases, ATPases also get affected, because of which the electrochemical gradient will get affected. Then the membrane phospholipases. So, evidence of specific high affinity elicitor binding site, as I said, in plasma membrane is available. Inhibition of the plasma membrane ATPase that affects the proton electrochemical gradient across the membrane. Therefore, the signal transduction chain between the elicitor receptor complex causes the gene activation process. One of the ways was your calcium cyclic AMP pathway. The signals are either transported locally by diffusion through intracellular or extracellular fluids, infection sites, or they can be systematically transferred through your xylem elements, which you remember we were talking about when we were discussing secondary metabolism in plants, that salicylic acid, ethylene or jasmonic acid were formed near the point of infection, and then the signals were sent through xylem to other parts of the plant. So, what is important when we are optimizing elicitors, or when we need to optimize what kind of elicitor, what concentration? Elicitor specificity. if an elicitor is working well or leading to maximum enhancement in a particular species, there is no guarantee that it will work well with all the other species. So, there is elicitor specificity involved. Elicitor concentration is one of the factors which has to be optimized. Duration of elicitor contact is crucial, because these being elicitors, they are affecting the cell viability. So, it has to be taken into account the exposure time. Maybe if you expose it for a longer duration, it may affect the cell viability and growth of the culture, in turn leading to lesser productivity. Even if, as I was discussing in the earlier classes, the ultimate aim is to maximize productivity. Then growth stage of the culture, at what stage? So, this indirectly involves the time of addition of elicitor. So, elicitation of cell suspension cultures can result in the following things. Different products may show high levels of accumulation at different times. If there are more than one products forming, it is not necessary that all the products will get induced at the same time. So, new product accumulation which might happen, as I said, I gave you an example of cyclotides. Some of the novel cyclotides came up in the in vitro cultures, could be because of the stress under the in vitro conditions, because you are growing them in a defined media under controlled conditions, which could have been a stress to the plant cells, leading to induction of certain genes or expression of certain genes which were lying encrypted under normal conditions. Then excretion or leakage of the product due to cell breakdown. It is not recommended that elicitors should be used for product secretion. You have set strategies. What was that which we studied in the last class? If you want the product to be out. Cell permeability enhancers. Elicitors may lead to breakdown, but then that leads to compromise in the biosynthetic potential of the cell. The growth rate will be compromised, or the viability of the cell. Now, what are the different types of elicitors? One, which are originated outside the cell and which induce immediate reactions, and these endogenous elicitors which are formed by secondary reactions, which means once the infection has taken place outside, then the subsequent changes or the signal transduction may be, which happens, also form the class of secondary metabolites. So, these are called as endogenous elicitors, and what will impact the first, the cell would be, or exogenously would be exogenous elicitors. Now, even in that, there are two classes which I referred. One is biotic elicitors and the other is abiotic elicitors, depending on their origin. Now, biotic elicitors, these are directly released by microorganisms and recognized by the plant cells, enzymes, cell wall fragments, then formed by the action of microorganisms on the plant cells, like for example, fragments of pectins can be, then formed by the action of plant enzymes on microbial cell walls. Then you would end up, if it is a fungus, then you can see chitin, then or carbohydrates, cell wall components, then compounds endogenous and constitutive in nature, formed or released by the plant cell in response to various stimuli. Abiotic elicitors, they can be of physical or chemical in nature. They influence endogenously formed biotic elicitors. So, the abiotic elicitors like physical factors, UV light can be one, then your freeze, suddenly you freeze and thaw. So, freeze thaw cycle can be a stress. Then medium components like agarose, agrigel, like for example I told you, once you bring in the culture under in vitro conditions, you are growing it in the presence of such components, which may cause an impact on the secondary metabolism. Now, chemicals which can disrupt membrane, for example your detergents, fungicides, herbicides, so anything which can cause stress, but it is of synthetic nature, then that forms abiotic elicitors. Now, biotic and abiotic elicitors, they differ in their dose effect relationship. What does that mean? Which means what? What is needed? That is the reason why concentration optimization is necessary. At what concentration it is, you cannot generalize. Every biotic or abiotic elicitor, because they are specific, they are concentration dependent, exposure time dependent, so optimization is necessary. Not all enzymes, as I said earlier, will get affected by a particular elicitor. A particular elicitor might be affecting one particular enzyme, another elicitor might affect some other enzyme in the pathway. Then concentration of the elicitor used is critical for maximum effect on the cultures. Product accumulation, generally what is observed is as you would increase, the product yield would increase, but beyond a certain concentration it will decrease. Why? Saturation. Right, because it is balancing between the yield enhancement and the toxic effect on the cell. So, what are biotic elicitors? Which originate in the invading organism, in which case they are referred as exogenous. Endogenous elicitors, which are biotic in nature, can be of plant origin and are generated by interaction between the microorganism, which means the pathogen and the plant, which may be plant components. Then biotic elicitors, for example, polysaccharides, glycoproteins, lipids, lipopolysaccharides, oligosaccharides, so any active component which is coming from of biological origin, if endogenous is coming after the attack from the plant cell itself, or if coming from the pathogen, it is exogenous but a biotic elicitor. What are abiotic elicitors? As we said, diverse collection of molecules, they are not derived from natural sources such as tissues of pathogen. These are from natural sources of biological origin, but abiotic, for example, salts of heavy metals, these are very frequently used in literature as abiotic elicitors. Molecules involved in signal transduction pathway, if synthetically you exogenously add them, they are considered as abiotic elicitors. For example, salicylic acid, jasmonic acid, if you add them exogenously. Then detergents, factors that can cause stress, physical factors, repeated freezing, thawing cycles, wounding, creating wounds, then exposure to UV light which can be a stress, temperature shocks which is heat shock,